All right, no questions. Let's move on. I got lab 6 graded. So take a look at that. And lab 7, I think there was a problem with the time for submitting it. And a lot of you
Yeah, I do have
emailed it to me. I think what happened is when I rolled the course over from last term, every once in a while, a little bit of a bug or change happens, and it switched the time from 11:59 p.m. to 12:59 p.m. So the submission time was supposed to be like a little after noon on Saturday instead of 11:59 p.m. And if you guys let me know that problem in advance, I can correct it. But I didn't know of it until after the assignment was due and people were trying to turn in assignments. So I got it corrected now, and that'll be good for next term, of course, but it didn't help you guys. So I will get that graded and then open it up, and you can submit it once I get it opened up again.
I do have a question. It shows that there's like 3 lectures, 2 labs, and 3 quizzes this week on top of the project.
I don't know how you saw that. So I think you gave me an email and I looked at the
Somebody else
syllabus.
gave me an email, but I looked at it.
Uh-huh. And I told you I saw that there was only 1 quiz this week. Of course, it's in 2 parts, A and B, and 1 lab. I'll show you the syllabus in a minute. Let me go on. I've been working on the unknown notes. A lot of you guys haven't been working on your unknown projects. I'm going to warn you that this project will take you time. So you should get started on the unknown notes or the unknown project. Otherwise, you won't have time to get it done. I got quiz 1A graded, and I think I'm working on quiz 2A. I think that's all I need to talk about that. Let me go ahead and shut this down, and then I can show you the screen. Where do I go here? Just a second, trying to figure out there. Click on that.
Okay, because on the grading part, it says quiz 5 is also due by tomorrow at 8 p.m. Because there's quiz 4A, 4B, and then 5.
Let me show you the screen here. I don't know where you're reading that from.
It's on my screen, the grade book. If I could share it, I would.
Just a minute. This is the syllabus.
Okay, but what's different on the syllabus isn't the grade book though.
The grade book. Let me take a look at the grade book in a minute. In the syllabus, which is what we follow, we have quiz 4 this week and 1 lab 8. And we're supposed to be starting microbial genetics, but we're not going to get, well, we need to finish chapter 6 first, and I don't know if we'll get to chapter 8 or not today. We're behind schedule. And of course, we're also working on the infectious disease project. Its paper is due this Saturday, and you're working on your unknown project in the lab. So this is what we're doing. And let me take a look at the grade book, which I don't know why you'd look at that for the schedule, but let me shut this down because I'm not going to show you everybody's grades. Stop share. Quiz 5. I'm not even seeing quiz 5 in the grade book. There it is, quiz 5. And yeah, its date isn't correct. I need to change that. Okay. I'll do that later. There we go. When is quiz 5 supposed to be? Week of March 1st. So it's way off. I don't know if the times are correct on this quiz, but yeah, I'm correcting the date now. No, the time is right. 8 p.m. Okay. All right. Let me close that and get out of the grade book. Thank you for pointing out the problem in Canvas. All right, let's return to the lecture. Are you guys seeing the screen? Hello? Can you guys see the screen?
No, we can't.
Okay, thank you. All right, so today we're supposed to be starting chapter 8. We're not there, we're behind schedule. We need to finish chapter 6, and we're only about halfway through it. In the lab, there will be lab 8 today, as well as this week, your unknown project and the infectious disease. Let's move on. So I was talking about the 4 toxic forms of oxygen last time. And we talked about the first type being singlet oxygen, and the second type being superoxide free radicals. They get corrected with sod. Let me save this. The third type is peroxide anions, sorry. And this is an oxygen molecule with 2 extra electrons. 2 peroxide anions can combine with 2 hydrogen ions, forming oxygen and hydrogen peroxide. Hydrogen peroxide is a problem, but it's neutralized by the enzyme catalase. And you can take 2 hydrogen peroxides in the presence of catalase, and that creates 2 water and 2 oxygen molecules. This is the main way our cells break down hydrogen peroxide, and the way that many cells that live in oxygen break down hydrogen peroxide. Hydrogen peroxide is a toxic substance made, especially in aerobic respiration, and cells have to be able to deal with hydrogen peroxide. Another way that cells, particularly some bacteria cells, can break down hydrogen peroxide is combine it with 2 hydrogen ions in the presence of the enzyme peroxidase, making 2 water molecules. Any cell living in oxygen must produce either catalase or peroxidase or both. Otherwise, the cell cannot live in the presence of oxygen. Any questions about any of that? And then the 4th toxic form of oxygen is a hydroxyl radical. The radical is any atom or molecule that has an extra electron, and the OH hydroxyl can take an extra electron, making the hydroxyl radical. This is different from the hydroxide ion, which has a negative charge. This has no negative charge, meaning there is no extra electron at it. It just has a single free electron that likes to bind, pick up another electron. And it's also different from a hydroxide group in like alcohols, the OH, which has no extra electron or a single electron group. And what's happening here is that this electron is either paired with the electron in hydrogen, or the other electron will be paired with whatever is on this side of the hydroxyl group. This electron is not paired, and that's why it tends to grab another electron. The singlet oxygen or singlet electron doesn't like to be alone, and so this will grab another electron. And that makes a radical a problem, and that is whenever this molecule bumps into something else, it'll steal an electron from them. And then that can do damage to the molecule that it steals the electron from. Hydroxyl radicals are formed by aerobic respiration in trace amounts. They're also formed by ionizing radiation. And then when hydrogen peroxide reacts with certain metal ions. Our cells need to react and remove these hydroxyl radicals as well as any other radical. And for the hydroxyl radical, we have antioxidants to remove this and other free radicals. 2 common antioxidants that we have, and I'm sure you've heard of, which also remove other radicals, are vitamin C and vitamin E, and then beta carotene as well. They're antioxidants, and they'll combine with the hydroxyl radical and then neutralize it. Any question about any of that? So oxygen is toxic even to cells that perform aerobic respiration. And it's not oxygen itself, but 4 oxygen derivatives, which are toxic. All microbes that use oxygen are called aerobes, and they produce more ATP from nutrients than those that do not use oxygen, and they're called anaerobes. But all organisms must have some way of dealing with the toxic forms of oxygen, otherwise they would die. And as you can imagine, obligate anaerobes have no way to neutralize or deactivate the oxygen toxic molecules or derivatives. And so they do not grow in oxygen. They will be killed by oxygen. We can classify microbes based on the effect oxygen has on their growth. Let me blow this up. So if an organism is growing in a thioglycolate tube, thioglycolate sets up an oxygen gradient. At the top of the tube, you have full oxygen, so that's 20 to 21%. And as you go down the tube, you have reduced oxygen. And then at the bottom of the tube, you have no or very little oxygen. So the obligate anaerobes will only grow in a thioglycolate tube at the bottom of the tube. And as you go up the tube, you'll see less and less growth. So you should actually see right here, 2 dots and then 1 dot. So as you go up the tube, you see less and less growth, because as there's more and more oxygen, the oxygen is toxic to these cells, they will not grow. The opposite is the obligate aerobes. You only see them growing at the top of the tube where there's complete oxygen. And as you go down the tube, there should be like 1 right here and 2 right there, and 3 right there. As you go down the tube, you see less and less growth because there's less oxygen the further down you go in the tube. And then at the bottom of the tube, you see no growth because there's essentially no oxygen. Now, if you grow organisms in the tube and you have the most growth at the top of the tube, and then as you go down the tube, you have less and less growth, but you have growth everywhere in the tube. Just that as you go down the tube, you have less and less growth. This is called a facultative anaerobe. These are organisms that can grow aerobically, and they grow best in the presence of oxygen because they're using aerobic respiration to make ATP. But if oxygen isn't present, they can switch to some other form for making ATP, like anaerobic respiration. So they will grow everywhere in the tube. It's just they'll grow best at the top where there's full oxygen. There's also organisms which we call aerotolerant anaerobes, and they grow uniformly everywhere in the tube. They do not use oxygen to grow because they're an anaerobe. However, they have all the enzymes and other molecules they need to remove the toxic forms of oxygen. So they can grow as well in the presence of oxygen as they can in the absence of oxygen. Any questions about that?
Is that why for sepsis they do 2 different tubes for the blood culture?
I'm not sure of that test. Are they growing it in thioglycolate?
I don't know. They always have a straw 2 tubes for the blood culture. That's why I was wondering if it was maybe both different types to find out what bacteria it is.
Well, I'm sure that it's a test to determine what bacteria it is. However, I do not know specifically the answer to your question, so I'm not going to answer it because I don't know.
Okay.
All right. There's one more group, and that is if you grow the cells in a thioglycolate tube, and they have the best growth in the someplace in the center of the tube, and less growth as you go up the tube, and less growth as you go down the tube. These are microaerophiles. They are aerobes, so they must grow in the presence of oxygen, but they prefer oxygen at less than 20 or 21%, so they do not grow as well at the top of the tube. They grow best in the middle of the tube where the oxygen is generally between 2 and 10%. The microorganisms growing in your stomach are microaerophiles. Helicobacter pylori, bacteria that causes ulcers in your stomach, is a microaerophile. Okay, it grows best when the oxygen is reduced, but not completely absent. All right, any questions about oxygen and the effect on microbial growth? All right, let's move on to culture media. There are different types of culture media. We've discussed this in the lab. A culture medium is the single word. So if you're talking about one, you use the word medium. However, if you're talking about many types of media, this is plural. So you use the word media. The medium can be a liquid or a solid solution. The difference is the solid has something added to the liquid to make it a solid, and that's usually agar, but there could be other things that are added as well to make it a solid media. It's the same nutrients. The only difference is it has something added to make it a solid. In the lab, you would be given a sterile medium, and that's one that has no living microbes in it. And then you would inoculate it with an inoculum. Those are microbes introduced into the media to get them to grow. And we call that a culture. Microbes growing in or on a culture medium. We call that a culture. There are a very wide variety of media that are available in microbiology to grow microbes. If we were in the lab, I would show you a book, and it's almost this thick. I don't know if you can see that very well. And it's a book that's about normal size, about this long, and each page has a different type of media for growing microbes. So that tells you there's a large number of pages for growing different media in microbiology. Any questions about that? A wide variety of media available to grow microbes. Put that down. Media contains all the nutrients that are needed for microbial growth. So media has everything you need to grow microbes, an energy source, a carbon source, and that can often be the same molecule like glucose. There's usually a source of nitrogen, sulfur, and phosphorus in the media. There's trace elements, and those usually just come from the media itself, like the powder you add to make the media, or they may come from the water. Trace elements, usually trace minerals, things like that, copper, iron. There may be organic growth factors if the microorganism requires it. And as I mentioned, an organic growth factor for us is vitamin C. Many bacteria do not require an organic growth factor, but there are some that do. And these, we usually supplement the media to supply the organic growth factors. What's the one that causes strep throat? Streptococcus pyogenes has organic growth factors, and they would not grow unless you supply the organic growth factors. And usually what we do for that one is we grow it on blood agar, and then the red blood cells supply the organic growth factors for the bacteria. All of these must be added to the media if the organism requires them. Otherwise, the organism would not grow. For most bacteria, you do not need to add organic growth factors, and we can just grow them in a general purpose media. Like E. coli would be one that'll grow in a general purpose media. I mentioned this briefly, that we can add agar to a liquid media to make it a solid. Agar is a complex polysaccharide derived from seaweed. And we usually we usually use agar in agar plates or Petri plates, but you can grow them in tubes as well. The tubes can be a slant or a deep. Let me just, see if I can get this quickly. Hopefully this will open and paint. So a tube would be where we have the agar straight across the tube, and that's not very straight, but you're getting the picture. A slant will be where we have the agar in something like that. Let me erase that. That went a little bit too far. And another slant, which has a bud, will have the agar poured first in a slat, and then the slant put on top of it. My lines aren't very straight today, but you're getting the picture. Oh, so a slant and the tube with the agar going across, we call that a D. Agar is generally not metabolized by microbes, and that's why we use it in microbiology to make our petri dishes. Edward Koch was the first to make solid media, and he used gelatin. The trouble with gelatin was that some microbes can digest the gelatin and then turn the solid agar into a liquid, and then that would make a mess of your plate. We have different media. The first one I'm going to talk about is chemically defined media. This is where we know the exact chemical composition in the media. Let me blow this up. Like this media, you make it by adding 5 grams of glucose, 1 gram of ammonium phosphate, 5 grams of sodium chloride, 0.2 grams of magnesium sulfate, 1 gram of potassium phosphate. And then you add 1 liter of water and you use distilled water. And then we know exactly what molecules are in the media. Many growth factors may be required to get an organism growing. Now, E. coli could grow in the first media, but many other organisms could not. And so we would have to add other ingredients. And this is also another chemically defined media, but you can see we're adding a whole lot of other factors, including growth factors over here. And if the organism requires a lot of things or additives, we call these fastidious organisms, ones that are not easy to grow. One fastidious organism would be Streptococcus pyogenes. It will not grow on a tryptic soy agar plate or a nutrient agar plate, meaning a general purpose media. Chemically defined media are usually used for growing autotrophs. And that's because they can make a lot of the molecules they need. They do not need a lot of nutrients to build their molecules. So autotrophs are easier to grow, and we frequently will grow them in chemically defined media. We also have complex media, and these contain extracts and digests of yeast, meats, or plant that provide vitamins and organic growth factors. With the complex media, we do not know the exact chemical composition of the media. Like for example, this one here, let me blow it up just a little bit so you can read it. It has 3 grams of beef extract. That will supply any of the molecules that were in the cow that we made the beef extract from. And that would depend on whatever the cow had been eating. So we don't know all of the molecules in a complex media. They do have a peptone source, which is a partial protein digest as the primary energy source. Frequently, they will have sugar added, although nutrient auger does not. The complex media usually supply a carbon source, a nitrogen source, and a sulfur source. The nitrogen and sulfur source could simply be the protein molecule in the peptone. We use complex media for growing heterotrophs, as these bacteria and fungi require an organic carbon source. And so we need to supply that in the media. And we do that by having a complex media. The simplest complex media are simply the general purpose media, something like tryptic soy agar or nutrient agar. Nutrient agar is actually shown right here. We also have anaerobic media. These media are important for growing obligate anaerobes. These organisms must avoid exposure to oxygen, and the anaerobic media is also called the reducing media. It contains chemicals like sodium thioglycolate that remove the oxygen from the media. Meaning the oxygen ties up the oxygen, and that results in oxygen depletion. We usually heat these media beforehand, and that removes the absorbed oxygen from the media. And so if oxygen comes back, it has to come in from the air, and then the top of the media will have more oxygen than lower down in the tube. We also have selective media. These are media that have a chemical that halts or inhibits the growth of a particular group of organism while not affecting the growth of the other group of organisms. An example of a selective media is 7% sodium chloride, and we actually call it 7% sodium chloride media. Most organisms cannot grow above 2% sodium chloride. So any organism that grows in 7% sodium chloride media must tolerate that 7% sodium chloride. And Staphylococcus species can grow on a 7% sodium chloride media, but E. coli cannot. So E. coli would be selected against. There's also differential media, and this is a media that provides visual differences between different groups of cells. The differences that we observe are not related to how well the organism grows on the media. The differences that you see make it easy to distinguish colonies of different microbes. For example, a blood agar plate is a differential media as well as an enriched media. We'll talk a little bit about that later. We can see 3 different types of hemolysis on a blood agar plate. We can have gamma hemolysis, that's no hemolysis, where the blood is red all the way up to the cells right here. Then you can have alpha hemolysis, where the red blood cells are partially lysed. And that's why we have the dark ring around the alpha, which is the bacteria, and why it's dark inside the alpha. From the partial hemolysis of the blood, it's darker. You might look at the plate and see it as a greenish color, but sometimes it's just dirt. We also have beta hemolysis, and that is where the red blood cells are completely hemolyzed. This organism shown here in the white is where the organism is growing. And you'll notice a clear region around it as well as inside of it. And that is from the complete lysing of the blood in the agar. So you can actually see through the plate right here. And it's better to call that clear rather than white. Any question about any of that? All right. There are different types of media and they can be combined. Like a blood agar plate is both an enriched media as well as a differential media. We also have selective and differential media like eosine methylene blue or EMB plates. They're selective against gram-positive bacteria, but they have ingredients that allow gram-negative bacteria to grow. And actually it's the dyes eosine and methylene blue, which are in the plate, which are making it selective. The gram-positives don't like growing in the presence of these two dyes. It gets in the cell and prevents the cell from growing. It's differential on the basis of lactose fermentation, meaning EMB plates have lactose in it. And if the bacteria can ferment the lactose, I'll blow this up, then they will be black or dark in color or a greenish coloration. And that's because the bacteria ferment the lactose, creating an acid, and that'll lower the pH. And when the pH lowers on EMB, we'll have a black color or a greenish color. That's different from other bacteria which do not ferment the lactose, and they will have a white or off-white appearance. They do not ferment the lactose, meaning a color other than black or greenish. We have another type of media that we call an enrichment media. It encourages the growth of some microorganisms to grow, but selects against other microbes. So it is a selective media, but it's a special selective media that we use to enrich for an organism of interest. It increases the numbers of desired microbes to detectable levels. It's particularly useful if you're looking for a microorganism and it's present only in a small proportion of the population. It's often used when we're looking for microbes from the soil or fecal samples, because the microbe which could be causing the problem, like a diarrhea, some other chemical, some other problem in the patient, and you're looking for fecal specimens to see if they have a pathogen, well, you're going to have a lot of other microbes in the feces, and you need to have some way for enriching for the microorganism of interest so that you can see it. For example, if the microorganism of interest was only one cell and 10 to the fourth cells, what is that, 10,000, something like that, and you streak out for it, you would never find a colony of interest for the microorganism you're looking at for. So what you do is you take the sample containing the few organisms of interest and then grow it in an enrichment media, which will encourage that organism to grow. Let's say you were looking for a phenol degrading bacteria, and you're looking at the bacteria in the soil, well, the number of phenol degrading bacteria in the soil will be very, very low. And you never find it if you were just going to streak for it. However, you can inoculate the soil bacteria in a culture where the only energy source is phenol. Now, only the organisms that can grow in the presence of phenol will grow in these tubes, and that will enrich the sample to a detectable level. What you do is you transfer 1 ml of the soil into the media containing phenol, and that will enrich for phenol metabolizing organisms. And then if you need to enrich further, you take 1 ml out of that enrichment media and put it into a fresh tube of enrichment media. And that way you could get the phenol metabolizing bacteria to grow, and that will increase its numbers. And once you get the number to something like less than 1 in 100, not less than 1, more than 1 in 100, then you would be able to streak and get it. Meaning it's possible to streak out on a culture plate 100 different isolated colonies. It'll take you some time and some work, but you could get it if you had the phenol degrading organism at 1 in 100. If you wanted to be lazier, you could enrich it further, meaning take it from that second enrichment media and put a sample of it in a third enrichment media and enrich further. If you were to enrich it so that you now have the phenol degrading bacteria, 1 in 10 bacteria, you would easily be able to streak it out and obtain a colony of the organism of interest. Any question about any of that? All right, this table tells you the different types of media. A pure culture is a tube or plate that only contains 1 species of microorganism. A colony is a population of cells arising from a single cell or a very few number of cells that might be clumped together. And then they form a colony as those cells divide and divide and divide, where 1 cell can grow into 1,000,000. And then once you have 1,000,000, you'd be able to see it as a colony. Colonies are composed of, at the very least, around 1,000,000 cells. But most colonies have 10 to 100 million cells. So to obtain single colonies is a critical part of obtaining a pure culture. You use the streak plate method, meaning streaking for isolation, to get isolated colonies. However, if you were to do this just 1 time, you would not get the purity high enough so that you wouldn't run into contamination or contaminants. So what you need to do is pass this colony, which is an isolated colony, onto another plate and then streak it out for isolated colonies. If you send cells through 2 colony isolation events, now you can pick from a single colony, and there will be a very good chance that that colony will be pure, meaning cells of only 1 species. And the purity will be at least 95% or higher, meaning if you were to do 100 colonies, at least 95% of them would be a pure colony. But you do need to pass it through two colony isolation events before you can call it pure. This is showing you the streak plate method, where you streak out the cells in quadrant 1. Initially, quadrant 1 will be heaviest here, but as you go and streak, it'll get lighter as more and more cells come off onto the plate. You then streak out and do colony quadrant or sector 2, and it'll be heaviest in this region. It'll pick up cells. Actually, you need to overlap with quadrant 1. So you pick up cells in quadrant 1 because you sterilize the loop. You do quadrant 2. And in reality, this should be moving. The start should be immediately picking up cells from quadrant 1. And then you sterilize your loop, and then you do quadrant 3. You streak out quadrant or sector 3. You'll note that you do not form a quad sector from the heaviest streaking of the previous sector. Meaning we're in the area where the bacteria are lower in numbers, as well as here. And that's to get further isolation or and deletion, not deletion, further, what's the word I'm looking for? Reducing the number of cells. It's not the word I was looking for, but you get the idea. Dilution of cells, that's what I'm looking for. So when you're doing this second streak, you don't go all the way up to the highest proportion of the cells. You should only go lightly into the cells to pick up the cells. And that way you're starting from quadrant 1 at the low end, not at the high end, to set up quadrant 2. And as you can see, quadrant 2 is starting to get isolated cells. And then you do quadrant or sector 3, and you sterilize your loop, and then you come into quadrant 2 to streak out sector 3. Any questions about streaking for isolation? Okay, most bacteria grow by binary fission. That's where one cell splits into 2. But there are a few bacteria that can grow by budding, where one cell forms an outgrowth that we call a bud, and that bud will enlarge and enlarge with time. It will eventually separate from the parent cell, usually when it's about the same size as the parent cell. And I'll show you a little video or pictures of that. And there's one other way that cells, bacteria cells, can divide, and that is by forming reproductive spores. See if I can get a picture again. Pull that up a little. Usually the parent cell will do something like that. And it'll form a few reproductive spores at the end. The reproductive spores are usually at the end so that the spores can be picked up by something or blown by the wind and then go to another location. Another good arrow. And then they will germinate the reproductive spore into a vegetative cell, which will then go on and perhaps reproduce by making reproductive spores. Any question about that? So binary fission, the bacteria replicates its DNA. One copy moves to one side of the cell, the other copy to the other side of the cell. And then the cell literally splits down the middle, growing a plasma membrane and cell wall right in the middle of the cell, splitting the cell into two. That's binary fission. And here we're seeing an actual electron microscopic image of a cell going through binary fission. Cells can also grow by budding. Let me see if I can get this to work. Where a cell will put out a bud, and that bud will grow, bud will grow, bud will grow, until the bud will be about the same size as the parent cell. Then it will break off from the parent cell. Any question about that? All right, where's the video? There, oh, there it is. I'm going. So we're watching this cell here, and there you can see it's starting to put out a bud. And there's the bud there. The DNA is starting to replicate. We're watching that there. And right there, the DNA is starting to move into this bud. And then the bud starts growing. It's getting close in size to the parent cell, and right about there, it totally separates from the parent cell. Now it's its independent cell, and it will go on and start forming a bud right here, as well as one forming here. And that will eventually go on and form a cell the same as this one did. Any questions about that? All right, when we're talking about the growth of the bacteria, it's important to talk about their generation time. This is the time required for a cell to divide. It's also the time for a population to double in size. They are the same time. So with humans, the generation time is something like 20, 25 years in the United States. Of course, it depends where in the world you are. And that would be the same about time as for the population to double. This varies considerably, the generation time between species. It can be as fast as 20 minutes. E. coli, when it's growing its most rapidly, can go through binary fission every 20 minutes. Most bacteria require a time between 1 and 3 hours. And then there's some which are very slow in their generation time, like Microbacterium leprae, which causes leprosy, requires 10 days for one cell to divide into 2 cells. Note that I state the generation time of like E. coli is 20 minutes. That's an abbreviation. It's understood that when I say 20 minutes, it means 20 minutes per generation. Students have a difficult time with the generation time because I guess maybe like from driving, we're used to talking about something like miles per hour, units of distance traveled per time. So you think of per time. But the generation time is not that. It is the units of time per generation. Any question about any of that? Microbiologists, you like to plot a bacterial growth on a semi-log plot, where we plot the number of cells on the log scale on the y-axis, and then the generation on the x-axis. this is normal arithmetic, meaning normal time scale. This is a log scale. That's why it's semi-log scale. When we use the semi-log scale, we get a very easy reaction to describe. Y equals mx plus b, a very easy way to describe the growth of the cells on a semi-log plot. If we weren't using a semi-log plot, we'd have this appearance, and that's much more complex. That would be something like y equals mx plus nx squared plus b, and then maybe plus another constant. And this is much easier to chemically describe as well as, not chemically describe, but to describe as well as to use. It's not quite as important today because of calculators being everywhere, but this would be a much more difficult equation to work with. Anything else I want to talk about that? Oh yeah, the other advantage of this is that it tends to plot out the points, I don't know, reasonably, that they're scattered throughout the graph. And then the next point will actually be, you can extend it by, it would be something like around here. So it's still on the graph, or you can extend the graph to it. Whereas here, the next point here would be on the ceiling, okay? And then all the points down here are all clumped together. One cell divides into 2, 2 into 4, 4 into 8, 8 into 16, 16 into 32. Now this would scatter them, okay, as shown here. And that would be whatever log 1 is, and log 2, and log 4, and then log 8, and then log 16, and then log 32. Anyways, the point is it scatters them better than the arithmetic plot. When we're talking about bacterial growth, it's important to talk about the phases of growth. Bacterial growth curves generally have this shape, where when you're growing a media from a liquid culture or a solid culture and then growing it in a liquid culture, initially, when you first put the bacteria in the culture, they don't grow. They're in a lag phase. What they're doing is they're making the enzymes they need to grow in this new media. Once they have the enzymes made to grow in that media, the bacteria can then grow. And that we call log or exponential growth, where the bacteria grow very rapidly. That will begin to slow and then enter the stationary phase. And why that's the case is the bacteria number have increased. Now there's fewer nutrients per bacteria. Waste products are building up, so they'll slow the growth. What's the third thing? Drawing a blank here. Oh, there's a shortage, starting to be a shortage of space. Initially, there was lots of space around. Now there will be a shortage of space because there's so many cells, not so much space. And then if we were to continue to grow the cells, the population would reach the death or logarithmic decline phase, where the bacteria numbers decrease. Initially, they would be in lag, and then when there's lots of nutrients, lots of space, very few waste products around, they grow exponentially. Then here, they're growing at the same rate as they're dying, and so the population is in the stationary phase, where the number of the population stays about the same. Here, early in the death or decline phase, You do have some cells, lucky ones, growing, but most of the cells are dying. And so the number dying are more than the cells growing, so the population number decreases. Late in the death or decline phase, all of the cells will be dying. There will not be any that are reproducing. And that's just that there are too many waste products around, not enough nutrients, not enough space. All populations tend to go through these 4 phases: lag, log, or exponential, stationary, and then the death phase. And that's true of deer and humans as well. We and humans have never even quite reached the stationary phase. We're still in the log or exponential phase. But if we start running out of food, we would enter the stationary phase. And then if there's not enough food, it would be entering the death phase, where the number of deaths are more than the number of births, or the deaths are the only thing that's going on. So you have cells that are stationary and you have cells that are dying. How long it takes the cells to die would be depending on the species. If it's endospores, the death phase would take decades, millennia. You may take a long time if it has endospores. All right, let's briefly talk about the ways we can measure microbial growth. There are different ways to measure microbial growth, and I've used all of them except for the most probable number. The plate counts, also called the viable plate counts, colonies on plates. There's the filtration method, the most probable number method, and the direct microscopic count. All of these are direct methods where we directly count the cells, or on a plate, the number of cells that form colonies. There are also indirect methods for getting a microbial growth. You could use turbidity. I'll talk a little bit about that. Metabolic activity and dry weight. Here we're using something and then we estimate the number of cells that we have. Plate counts are the most common method used for getting a direct measurement in microbiology. It measures the number of viable cells, so it's sometimes called the viable plate count. It does take 24 or more hours for the colonies to grow, so this one takes a little bit of time. And their results are often reported as colony forming units because we don't know if a colony was formed by one cell or if it was felled by a clump or a few cells. What you do in a plate count is first you make a serial dilution. You take 9 mils of media and you add 1 mil of your initial inoculum in, and that makes a 1 in 10 dilution. You then mix that up. And if you want further dilutions, you take 1 mil out of that, adding it to 9. And now the cells will be diluted by 1 in 100. That's 1 times 10 times 1 times 10, 1 in 100. And further dilution, you take 1 mil out of this 1 in 100, put it into 9 mils, you got 1 in 1,000. Further dilution, take 1 mil out of this, put it in 1 in 9 mils here. Now we have the dilution 1 in 10,000. And you can get further by taking 1 mil and putting it in 9 mils, making it a 1 in 100,000. You then plate each of these tubes onto a plate. Usually you only put an aliquot on, like either 0.1 mil, and then you have to spread it around. Or you can put on 1 mil of the sample and then move it around. You could spread it or just tilt the plate, or this one looks like they're rotating the plate. And then incubate these plates and count the number of colonies that form. And that will get you an estimate for the number of cells you have in your original inoculum. Now you have to plate media from each of the serial dilution. Many of the plates you cannot count. There will be too many cells to count, so you can't get an estimate on the number of cells in the original media. And then some will be too few to count. You want cells between about 30 and 300 colonies. Sometimes they say 25 colonies to 200, 250 colonies. And the reason is that the mean here is much more accurate than the mean here. The mean, if it's 1 in 30, means is that you're going to be off on your mean by only 1 in 30. Whereas here, if you're off on your mean, the mean looks like it's 4, and it's plus or minus 1. Well, that means you're plus or minus 25% of your count. And so we like to use the plate, the dilution, that gives rise to a plate where the number is between 25 and 250, or maybe 25 and 300. And that way you get a more accurate result, and the mean will be more meaningful. You can also do a filtration, and that's when the bacterial numbers are very low in number. You pass the bacteria through a filter. The bacteria cell will be lodged on the filter. You then put the filter on a plate of media and then allow it to grow, and you count the number of cells to determine the original concentration. And this would be kind of tricky because you took like 1 mil. Well, you could do it not 1 mil, but 1 liter, and you pass it through the filter, and then you get it to grow up. And this would be the number of colonies per liter. The filtration method is only used when your bacterial numbers are quite low and you can't get your colonies in another way. You can actually do a direct microscopic count. You have a special slide which has a specific volume underneath the slide. And usually what you do is you count one of these squares right here, and then that will be the 1/1,000,000th of your sample. And so you take the count here, what would that be? Something like 20 to 30, and then multiply by 1,000,000. The counting method, of course, is a little bit difficult in the sense that whoever has to do it, it's kind of hard on their eyes. And if the cell moved, it would be impossible to count because this one would move up here, that one would move down there. But the good thing about a microscopic count is you can actually get your numbers right away. You have your cells, you do the microscopic count, and then you have your estimate of the number of cells. All right, so that's it for the direct method. Let's talk about the indirect methods. You can use turbidity, where a light is shined through the media, and then you measure the amount of light coming through the media. And then you do this with the solution having the cells in it, and wherever there's a cell, it'll block the light rays from coming from the light source, and so you'll get less of a reading. And you need to then look on a chart where somebody has given you your numbers, and you will take a look at your turbidity, the measurement here. I don't know if that's about 20. And then look on the chart, and that will tell you how many cells you have. This one I really like because it's very quick. You just take the cells you want, put it in the turbidity meter, and then get the reading, look on the chart, and you know your number of cells. You can also look at, get an estimate of the number of bacterial cells by centrifuging, oops, that's this one here, centrifuging the cells and then drying the cells out and then weighing the cells, the dry weight of the cells. It's usually a method that's used for bacteria and fungi, but it's not satisfactory for filamentous bacteria because they've got a special shape and it just doesn't work well, or for organisms that have like a thick glycocalyx. The advantage is that you can weigh them at your leisure whenever you have time. You can also use metabolic activity as an indirect method of getting an estimate. This assumes the bacterial metabolic product, like if you're measuring CO2, accumulates in direct proportion to the number of bacteria present. So this one, the more CO2 you have, the more bacteria you have. On this one, the higher the weight, the more bacteria you have. Any question about these methods for getting bacterial numbers?
No, but I think we skipped slide 42.
Was that the one just going over a topic?
It was a two-part slide.
Yeah, no, I already talked about this.
Okay.
That's why I skipped it. Because we're behind schedule. Let me just briefly, we only have a few minutes left, talk about chapter 8. I think I'll only introduce the topic. So chapter 8 is looking at the genetics of microbes. This slide is showing you the major goal, learn the terms, be able to describe DNA replication, RNA transcription, and protein translation, and understand that there are different types of mutation. So initially, we'll look at the structure and function of the genetic material, looking at the flow of genetic information, how the information starts in the DNA, and that we'll look at can flow by DNA replication. And then that DNA information can be moved to RNA and transcription. And then the information in RNA can be translated into proteins in protein translation. Excuse me. We'll then talk about the regulation of bacterial gene expression. We'll talk about operon, so you need to know what an operon is. We'll talk about mutation. We'll talk about the genetic transfer. Like genes can move from one organism to another. And you know that obviously genes move from parents to offspring. We'll also talk about genetic recombination. And then lastly, we'll talk about genes and evolution. I think I'll just stop here and we'll pick up here on Thursday. We're still behind schedule, but oh, come on. Any questions?
So were you saying that you were reviewing our unknown projects today? Because I asked for a test to be ran on Thursday.
I will look at it. It depends how long the test incubates before you get the results.
Okay, thank you.
Okay. But I sent out results last night. All right, I'll say goodbye if there's no questions.